joining us. Thank you all for joining us this morning for this panel discussion to talk about the blueberry health issues that were observed um, in this growing season. To join me on this panel discussion, we have Lisa Deveter. She's a small fruit horticulturist working with Washington State University. Lisa, do you want to join us? And we have two more speakers. Um, Caroline Teasdale, she's the industry specialist berries with BC Provincial Government. She is joining us online. And we also have Dimitar Malau. He's a plant virologist with USDA, and he is working in Oregon. So our panel members uh, will provide their expertise and share what they have observed during this growing season on this topic, and they would cover their respective regions from Washington, Oregon, and BC. So to start with, I or just give a little bit of background about what was observed this year, and then we do have a poll question right after I just right after my talk, and then we'll talk with our panel members. So right in mid-May, we were Um, so right during mid-May, we were receiving calls from the growers regarding some symptoms that they were observing in their fields, particularly with the Liberty cultivar. And the symptoms that were being observed were blighted shoots, as you can see in these images. The blue crop and the draper were also showing a similar kind of symptoms, and more with Duke and Rika as well. So in mid-June, I had surveyed 22 fields and looked for these shoot dieback symptoms. And these are some of the fields that I surveyed. I had surveyed 4 fields of Liberty fields, and out of which there was like 35 to 100% of the plants in this on these 4 fields. They were showing those shoot dieback symptoms. Um, at each field, I had observed 30 randomly selected plants, and whatever the data that I'm showing is based off of those 30 plants from each field. With the blue crop, I looked at 5 fields, and there was this 0 to 13% incidence of those symptoms. With the Draper, 2 fields, Rika, 2 fields, and Duke, 4 fields, and again, the symptoms varied between 0 to 30% disease incidence. I mean, not disease, I mean the incidence of those symptoms. With 4 fields which had Elliott, Hardy Blue, Draper, and Legacy, I did not observe any of these symptoms that I showed earlier. But one common thing that I observed among all these fields was that all the plants that were showing symptoms, they were showing symptoms similar to the shock. But at that point, I have not sampled any of these and for testing those for shock or scotch. But I know Tom Waters has collected some samples from some of these fields that I surveyed, and he passed on those samples to the USDA virologist Dimitar, and hopefully he will shed some light on these samples. in this panel today. I also looked at one field where there was two cultivars that were being grown on the same field close to each other, a Liberty field and a Draper field. What you can see here is I looked at 30 randomly selected plants from that field, and on the bottom panel you can see the lesion length of the shoots presented in centimeters, and on the upper panel you have the number of affected lateral shoots. I mean the shoots that were showing those dieback symptoms. So as you can see here in the top panel, the number of those affected shoots kept on increasing during that 1 month observation period. Whereas if you look at the lesion length, there wasn't much of an increase. So it's only the number of shoots that kept on increasing during the that 1 month period from the mid-June to mid-July period. With the Draper, I have not observed a similar kind of an increase, or there weren't a lot of affected lateral shoots with the Draper, even though they are in the same field next to each other. So there is something going on with the Liberty cultivar for sure, based on the observations that I made this season. And so I think it's a good segue to stop and then hear more from our panel members. But before that, I want each of you to pull out your smartphones and scan this QR code. And if you can, there's a poll question, and the question is about, like, on a scale of 1 to 5, could you share like how important is this issue to you? And I would also request you to keep this that page open, because there is one more poll question that is coming up towards the end of the panel. And all these responses are anonymous, so feel free to let us know like the importance of this issue to us. So I'll just give you 1 minute, and then we'll go on and talk to our panel members. Good morning, Carolyn. I hope you would be able to share with us like what's going on with blueberries in Canada, and then any virus testing and any results that you want to share with our growers. That would be great. You can take it from here, Carolyn.
Thanks, Shakradhar. Just checking that you can hear me. You can. Okay. Thank you. So I will give an update on the situation in British Columbia. I wanted to start by saying that the spring of 2022 in BC, it was quite cool. It was wet, and our plant development was really delayed. We had a fairly cold winter, but we didn't see a lot of cold damage, with the exception of calypso fields, where we did see some distinct cold damage in low spots or areas where the fields had a bit more exposure. But what we have been observing in BC is an increase in the number of virus symptoms over the last number of years. And This year was no different. However, I think the symptoms looked a little different than the classic symptoms than we've seen in the past. So I thought I would show some pictures. This first slide is not a picture from 2022. It's one that we typically use when we're trying to talk about virus and educate growers on what the symptoms look like. But on the left, you'll see that the virus symptoms that we typically talk about, we're talking about green stems with dead leaves and dead flower clusters. And then on the right, where we're seeing the brown tissue next to the dead flowers or leaf clusters, that's usually bacterial blight. However, I put this up because I think this year it was not quite so clear. And in some varieties, you actually do see blackened stems with scorch virus as well. So I will show some symptoms. What I wanted to start out by saying is that in BC, the virus situation, it's really variable from farm to farm. So we can't make general statements and say that it's all fields in one region or that it's all one variety. It really seems to be dependent on farm level management and probably the age of the plants. And so we have quite a few fields where we'll just have, one bush here or there that are showing symptoms. And I'm sure you're familiar with this as well. It's a typical pattern with virus we see anyways, is that it's not all the bushes in one row. It usually is one bush here, one bush there, sometimes a few together here and there. I thought I would go through, these are all pictures from 2022 that I'm showing here. So I thought I would go through the varieties just to show you, these are plants that are confirmed to have scorch virus. So this is what scorch virus is looking like for us. In Duke, we're seeing these sort of smaller leaves, some dead flower clusters, some dead flower clusters, some dead leaves, but typically these bushes, the leaves are smaller and yellower. It can vary though from field to field. You see here is a much more mature Duke and you're seeing a lot more brown on it. Close up of some Duke. This was one field that was unusual for us. So if there's some things that were unusual for us, it was that there were fields this year that had widespread symptoms that the growers were reporting they hadn't really seen the symptoms that much before. And I think these were fields that were probably have had scorch virus for many years, but for whatever reason, the symptoms were more obvious this year. And it may be because the plants were more stressed coming out of the 2021 really hot summer with water stress, as well as the flooding in the fall of 2021. So this was a Duke field where almost all these bushes in this one row were showing signs of scorch virus. In Elliott, we've always talked about Elliott as being one of the more obvious ones because the plants tend to go a little bit reddish. Again, lots of dead flower clusters and leaves. But one thing that's unique about Elliott, and I have seen this on Liberty as well, is sometimes you get these blackened stems. So this is what we wouldn't typically call a scorch virus symptom in other cultivars, but in Elliott, it's common to get this blackening of the stems as well. But the reddish tones on the leaves is a really clear indicator to us that it's virus. Another Elliott. Draper is usually pretty obvious as well. It can just be one branch or two branches on the bush one year, and then more of it goes the next year. Here's a bush that's a little bit further along, that's Draper. Now, one of the surprises this year is we saw an increase in number of symptoms in Rika fields. And so I don't know why, but it seemed like Rika was showing scorch virus symptoms more this year. And in the bottom right-hand corner on this slide, you can see we were also seeing this unusual reddening in the leaf veins in Rikas that had scorch virus symptoms. I don't know why, but it was something we observed this year. And then the other unusual thing this year was that for the first time, blue crop was really obvious in its display of virus symptoms. So blue crop, we've always talked about it as being the variety where it kind of masks the symptoms of scorch virus. The leaves turn slightly yellow. The bushes tend to be a little bit more upright and twiggy, but they don't die from scorch virus. This year we were seeing big patches of fields, of blue crop fields that were showing symptoms. So in this picture here, you can see the orange flagging tape is marking all the bushes with symptoms. These ones in the front, you can probably tell, slightly yellower. And they were even showing leaf dieback this year. So that's not something we'd really seen before. Again, you look along this row, you can see these first few bushes are quite green, and then things start to turn yellow. So that's blue crop with scorch. I did want to touch upon the history of scorch virus in the US and Canada because it's not new. It was first found in New Jersey in the 1960s, in Washington in 1980, in BC in 2000. So I think one thing that might make BC fields a little different than some of the fields in Washington is we have more of the older fields, particularly older blue crop. This virus has been here for over 20 years. So it has been spreading, and it has been spreading probably unnoticed. And you do get to a point of virus levels where all of a sudden it's really obvious that you have it, and it can look like something that we might call flooding damage, or whether, because it's so widespread by the time some growers are observing it. In BC, one of the things we've done since the early days of scorch virus, I think this actually started in about 2008, is we've had a virus testing program for our growers, which means that growers have been able to submit samples from their plants that are showing symptoms to the lab to have them tested for a scorch and shock virus. And this is free to them. So from 2013 to 2020, we had a program where we offered 10 free samples per grower, and you could see we had over 700 samples every year. Starting in 2021, we increased the number of free samples per grower up to 30. So you'll see like a tripling in the number of samples tested in 2021. That's not necessarily because there was this explosion in symptoms, although there were more and more symptoms showing up, but it's more because we were offering free testing for 3 times as many plants. And so this past year, we had the most samples ever with over 5,000 samples submitted. And these were submitted by over 200 farms. So 243 farms in BC submitted samples this year. So we're really getting the word out to our growers that this program is available. They should be testing to find out what's going on so that they can make informed management decisions. I wanted to end with some key observations. One is that great variability in virus levels from farm to farm. We in BC are getting a lot more bushes testing positive for scorch than for shock. So when you talk about shock-like symptoms, I mean, we talk about scorch-like symptoms because it's the predominant virus for us. Most of our scorch detections were in Duke, Blue Crop, Elliott, Rika, Draper, and Liberty this year. I think Duke, Blue Crop, and Elliott makes sense. They're the top 3 cultivars, but we're seeing a disproportionate number of scorch samples and scorch positives coming from Rika, Draper, and Liberty, like more samples coming from those than proportional acreage that we have. We're also detecting scorch in almost every cultivar, and the new cultivars like Calypso, Valor, Top Shelf, Cargo, Last Call. Our BC Blueberry Council, which is our industry association, has been emphasizing best management practices. So I just wanted to put them on the screen. That is to monitor your fields for virus symptoms, to test the bushes with symptoms, remove the infected bushes, don't let them regrow, take out the root ball, and then control aphids diligently, because aphids are what spread this virus. Wanted to end with a picture of one of our older blueberry fields where the grower is doing the right thing and cutting out the bushes with symptoms. Thanks.
Thanks, Kathleen, for sharing what's going on with the blueberries in BC. Now we'll move on to Demeter. He will share some of his experiences from Oregon State. Demeter, are you online? Demeter?
Hi, do you hear me?
Yes.
I couldn't hear initially when Caroline stopped what you were saying. Can you please repeat?
Do you have something to share, Demeter, like screen? Or do you want to talk?
I do not have a screen because I was not asked to prepare anything. I could have, but it's
fine, yeah.
But I can share a few things that we have been doing this summer. This is an issue that it's not just unique to British Columbia, but also, as you very well know, in Washington and all the way down to Oregon. And you did mention that we work with some of the growers in Washington, and that's true. We have not a large number. Hopefully, we can increase that, but we have a couple hundred samples from Washington this summer, and I can share that about 85% of those were positive for shock. Obviously, we're targeting mostly samples that you saw on the presentation from British Columbia. But then we have this idea and notion that shock recurrence, and we ran out of the hypothesis with the new luteovirus. Perhaps most of the growers and many of you are aware that there is a new luteovirus infecting blueberries, and some people speculated the virus is basically not causing major issues, but we want to investigate if you have infections with the luteovirus, can that actually make the shock recur, and perhaps does it affect the shock symptomology, and how does it work? So we did run samples, and we have for shock and luteovirus, and we have a large percent, actually, probably 60-some% were positive for both the luteovirus and shock, which we're investigating for 2-3 years just to see how this trend is going to go. But the number of samples that came positive for luteovirus and negative for shock is very minute. It's like few percent points only, and the majority of the samples from Washington were positive for shock. And for Oregon, actually, we did a much larger collection. We have 1,700-1,800 samples, and we're still processing those, but the percentage is very similar, about 70% positive for shock, and at least 1/4 of those are positive for both viruses, and 30% are positive for the luteovirus. So we're still processing Oregon samples, and as I said, we want to do that for at least 2-3 years to have a better understanding and better picture of what's going on. After we process all these shock and luteovirus samples, we're going to actually run for scorch all these samples too, just to add to the picture and see when we scored in the field, if symptoms are there, what kind of a test we get in the laboratory. So hopefully by next year conference, I can provide much more, informative summary and what we observed for 22. But for now, I think it's just data gathering. Perhaps most people don't even know of me. I started last year, summer of 21. So it's pretty much my first season is this past summer. And hopefully with the next couple of years and talking about collaboration with Chakradar and other people, hopefully we can gather more data that would be useful to provide more insights on this blueberry decline. So I think that's all I have for now, but happy to jump in later and answer questions when they're.
Thanks, Dimitar. Before we move on to Lisa, I have one more quick heads up. After Lisa's talk, there will be a poll question that will be coming up, and the question is about what do you estimate is your yield loss because of this issue? And again, your responses are all anonymous. So moving on, Lisa, do you want to provide your perspective of this issue that we observed this year, more from a horticultural perspective? I think that would be great.
Well, first, thanks to Chris Benedict and Chakradar Madapali for organizing this. I had the opportunity to visit some of these affected fields with Chakradar, and it's a really unfortunate and, from my perspective, a complex situation that's going on. So a lot of what, from my opinion, of what we're saying here is largely speculatory, and just like Dimitar said, We still are in this process of data collection and trying to figure out what has exactly happened within the Pacific Northwest region, because it could be due to biotic issues such as virus, could be due to environmental issues. We had a really challenging 2021 season with the heat dome that affected a lot of our fields within the region. But then we had a really cold, wet winter, and we also had some spring freezes as well. So again, a lot of it's speculatory. Some of these things can act in isolation. Some of these things can also act in combination, have these interactive effects that make it hard to tease apart. So I do want to comment a little bit about some of the observations that we made and how they relate to the environment. So we did have that heat dome in 2021. A lot of growers were affected. Hopefully not many of you within here, but what the heat dome could have done is that there could have been some vascular damage in the xylem. That's the pathway that conducts water and nutrients that are dissolved in the soil water solution. Essentially, there could have been embolisms that have developed. We might have not have noticed any symptoms, but later on that damage might have manifested with some stressed plants with our spring weather conditions. That's a possibility. I'm not saying that's what happened. We had some observations that suggest that didn't happen in all of our fields here in Washington. For example, Aurora was particularly hard hit with the heat dome, but we didn't notice Aurora more strongly affected by these spring conditions that was just described by Carolyn and Chakradar. Regarding cold and freeze damage, so some sites did have some vegetative damage to some spring frost events. We look closely at cold hardiness and freeze damage in my lab, and we've collaborated and developed cold hardiness models. We looked closely at those models, and we didn't see a consistent pattern that related to cold or freeze damage to what we were observing in the spring. For example, one particular field site that was extremely damaged, it was a Liberty field in Skagit County by this springtime condition. There was no indication of frost damage there as well. So again, it's complex. We really have put our heads together across the region and across different disciplines. And we're just at this point thinking it could be environmental, biotic with virus, or an interaction. So I again want to thank the region and all the people that have worked in the region for this coordinated and collaborative effort to try to pull together and figure out what's happening here. So that's all I have to say.
Thank you, Lisa. I think the poll questions are now open. If you can answer that question, that would be great. And as you can see from the three speakers that we have this morning, what we are trying to do with this panel discussion is we are still trying out the reason that cause for this phenomenon that we have observed in this growing season. We don't have any answers as of now, but the point with this panel discussion is to open up and then have a discussion and discuss the various possibilities so we can figure out a course of action. So at this point, if any of you from the audience, if you would like to add to what the speakers have already said, or more from your field observations or any such things, if you would like to share, that would be great. Yeah.
Good morning, everyone. I'm Christian, and I want to share some not studies, but we observe, during our varieties, our performance. Yeah, we got some cold and freeze damage, first of all, and if we can, what is the best argument for us? Because we have some devices to try to monitoring, the activity on plants, and specifically in photosynthesis, and we observe decrease on 30% in all the varieties during the springtime. This is the first observation, and obviously it's a relationship, a correlation between that observation and data with the early stage of growing, and mitosis, and finally the cell division. for us was clue this issue because we had based on our counting, we project at least in all the farm was 7 million pounds. And finally, we got just 5.2, 5.3. That means we have a projection decrease in 35, 45%. This is our limitation.
Thank you.
Thank you.
Thanks for sharing. Yeah, I think that's 35% yield loss is like a big thing. Hopefully, we'll be able to find some answers in the near future. Moving on, I think that I have a couple of questions that I would ask the panel members to continue on the discussion. My first question is I would like to start with Lisa. Can you describe like what the conditions for 2022 that would create this widespread symptoms?
Yeah, sure. Okay. Sounds like this microphone is on now. So I just will comment about photosynthesis. Photosynthesis was likely reduced this spring because we had a lot of overcast conditions and it was cold. So we weren't at optimal conditions for photosynthesis this spring. However, in the spring, the plants are largely using reserves that they've stored from the previous season. So we didn't really, from a horticultural or physiological perspective, think that photosynthesis this season led to any of the symptoms that we observed this spring. But I'd be happy to talk more about what your observations are and kind of separate these apart a little bit more and gain a better understanding. So with your question, Chakradar, so stressed plants from the preceding environmental conditions that we had in 2021 with the heat dome, like I mentioned, as well as our cold, wet winter, those could have led to stressed plants that could have been more susceptible to virus if there was already some latent infections from some of the viruses that Demeter and Carolyn mentioned. But again, largely speculatory on my part. So this is just based on observations that we've seen in the current information that we had on the region.
Carolyn, do you have any thoughts on why we are seeing this widespread symptoms this season?
Yes, well, I can't speak to your situation in Washington and Oregon because I haven't seen it firsthand. But in BC, I mean, we know what we're dealing with for the most part. We're getting positive scorch virus test results from bushes with symptoms. But I agree with Lisa. I think that the extreme weather and the stress on the plants has probably contributed to either display of symptoms or even maybe a change in the latency period for the virus. So with scorch virus, there's typically 1 to 2 years between when infection occurs and when the bushes show symptoms. The other piece is just as bushes get older and as virus spreads over time, we should expect more and more year after year unless fields are being removed and replanted. So I think it's my perspective is perhaps a little bit different because we know what we're dealing with in BC.
Thanks, Caroline. Demeter, do you want to share like what's it's like from Oregon?
Let me see. Actually, I didn't think. I tried to put my camera, but apparently I'm not allowed to. So anyway, I didn't realize the first time I talked to you that I didn't see me and you still can't. I guess there's some connectivity issues, but yes, what Lisa and Caroline said, I concur with these observations. The fact is that I'm a virologist and perhaps you need plant physiologists to really dabble on that. I have been talking to some of the physiologists, colleagues. Maybe the best approach would be not to speculate and kind of try to guess what it is, but going forward is to try to emulate all these weather conditions in a mini environment and combine those with different type of viruses, and perhaps that would be a good approach to go forward. So we know that when you have events like this, maybe we need to be prepared. But one thing I need to mention here is that there are a couple of viruses that were not really in the grasp of understanding what's going on in terms of epidemiology and how they're spread. Perhaps the weather events can influence potential vectors of these viruses, and maybe the higher incidence could be described by that. But again, this is purely speculative because we have no data. We're trying to see if we can do some traps, perhaps see if we can catch some aphids, and are they going to be positive for these viruses? This is very, very kind of in the infant stage. I don't know if I want to bring that here, but since you put me on the spot, we're also looking at other pathogens. You very well know Virginia Stockwell, who is my colleague here, and she and I are looking at some orchards that are declining, and unfortunately we're finding more pathogens. Where or not they play a role of this massive decline is still unknown, but she and I have a plan to explore that in a little bit more depth next summer. We're also looking at some other things like nematodes and bacterial pathogens that we're finding. So again, I think it's purely speculative at this point, but we have a lot of data points and a lot of directions that we can go for. And hopefully we can get a little bit of better idea in the next couple of years so we can deliver that and make better plans. But for now, I think what everybody is saying, I can't disagree with you, but we need more time and data.
Thanks, Dimitar. I would like to follow up with Dimitar's comment about the pathogens. Like I collected samples from some of the fields, and I tried to isolate bacterial and fungal pathogens from those samples. And I was not able to consistently isolate one particular pathogen from these samples. So we are still unsure of if the symptoms that we have observed this season is due to any bacterial or a fungal pathogen at this time point. So now I think we have enough discussion on this topic. I think the next thing that everyone wants to know is like, what should happen next moving forward? Like, what are the things that we should focus on? So probably we'll start with Lisa. She can give us her perspectives from a horticultural viewpoint.
Yeah, well, thanks again, Chakradar. Well, first, I think we just need to continue collaborating across the region and across disciplines so that we can observe and be coordinated in our observations and our strategies moving forward on how to address this. I do want to acknowledge Eric Gabrant in British Columbia. He has been making an effort to bring us all together since last year to discuss virus in particular, given it's such an issue in BC, and we have some indication that it's also a potential culprit here in Washington State. I also think we need to consider greater attention to virus research here in Washington State, such as testing and optimization of detection assays. In addition, I think that, perhaps we want to come up with a list of labs recommended for testing, because my understanding there's variation in lab results. And so maybe we want to make sure that we're giving our best recommendations to growers in terms of where they send their samples and make decisions based on those results. A lot of this would be an investment on the part of industry too, if we do research. So I think that discussion and coordination and strategizing has to happen with industry and some of our commissions as well. The other thing too, I was, really surprised when first engaging in some of these discussions with Carolyn about the range in symptomology that we see in virus. And I looked at some of our P&W handbooks, and it really doesn't go into all of the details of what virus looks like across the different cultivars, such as for scorch. So I think, communicating with growers, these are the symptoms to look for in Elliott. These are the symptoms to look for in Duke might be helpful so that we are better equipped to identify and proceed forward with testing and then making decisions based on those results. Then for those affected growers, our recommendation has been to remove some of that affected tissue that they've observed. And if they have experienced declines in growth and productivity, to prune hard this winter to try to stimulate regrowth. Some field sites actually regrew out of this situation quite well. Also, if they suspect they have virus, recommend that they follow through and conduct testing and then follow recommended practices if they do have positive detection of certain virus diseases. And lastly, I would recommend too that if they have questions, to contact their local area extension specialist across the region. Oh, and then I guess my last thing too is with the attention to virus too, I think we need to be more careful about vector management, particularly for scorch and management of aphids as well.
Thanks, Lisa. Caroline, do you have anything to add from BC perspective? Like, what are the things that you are planning to do in your region to handle this situation?
Yes, so in BC, we need continued adoption of best management practices for viruses by our growers. So that's monitoring, testing, removing infected plants, and controlling aphids. We also need more research to understand the rate of virus spread, the susceptibility of new cultivars, and to gain a better understanding of the virus and how it's behaving, or what other strains or other viruses might be doing. I think I wholeheartedly agree with you, Lisa. I think in Washington, you really need a virus testing program for your growers, and a coordinated virus testing program that, like what we found is a free virus testing program, works particularly well because growers need to be able to make informed decisions. It's highly likely you have scorch throughout Whatcom County. Aphids do not know international borders. And if it's scorch virus, then you don't want your growers pouring more investment into fields that are not going to recover. So really would emphasize the virus testing piece. I think it's just so much better to know what you're dealing with. And with virus testing, like, do that scorch virus testing at the same time as the shock testing if you can, because I think there's a good chance that it's a contributor. Yeah, that's all I'll say on that. Thanks.
Thanks, Caroline. Dimitar, do you want to add anything more to this conversation?
Yes, I think both Lisa and Caroline really sum it very well what's going on. I like the notion that we do need to be informed and we do need to know what we're dealing with. So testing is the key. I'm not a service provider, but rather my group here is doing research. And I think Lisa mentioned cooperation. So I can step up in a way that we're gathering a lot of data, and my short-term plan for the next 3 years is to test as many samples as we can. We're also trying to basically kind of dissect some of their genomic sequences and really make sure that the tests that we're using are very reliable, at least for the region here, because many of you with the recent pandemic know that could be challenging to detect viruses, and not all the time you do a test it's reliable and it comes positive 100% or negative 100%. So with that said, I think reaching across the border with Canada and across the border with Washington State, we can collaborate in terms of sharing, exchanging information, and each of us have unique capabilities, and we can contribute to each other's effort. So I'm committed to continue researching these issues and providing the best and the most up-to-date information I gather, so people can utilize it and hopefully use it to manage the issues across the borders. So that's how I see it for the next 2-3 years. And then once we get more data, 23, 24, obviously we can sit at the same table and discuss where we are and regroup and move again forward. So that's the way I see it.
Thanks, Dimitar. I think we are close to ending our session, and I just want to wrap up this panel discussion. I think we had a good discussion on this topic, and as you can figure out that we are still not sure like what the correct reasoning for the symptoms that we have observed this season, and we are in the researchers from BC, Oregon, and Washington, we are all working together to find out an answer for this solution. Meanwhile, if you have any questions or any suggestions regarding any of this issue or in terms of research, please feel free to reach out to any of the panel members. And before we close this session, I have one more poll question. This is the final poll question. We would like to know, like, if this panel has made any difference in how you view this issue. If you can input your responses, I think that would be really great. Yeah, I think Tom Walters would like to add something.
Oh, thank you, Chuck Rudder. I just wanted to add a couple of comments, particularly on scorch. I collected many of the Washington samples that Dimitra analyzed, and I'm really grateful for him for his interest in this important topic. I did want to emphasize that we have found several fields in Whatcom County that do have scorch. And in one of those fields, scorch was remarkably widespread. So scorch is here. It's not hypothetical. Maybe the aphids cross. We know that they cross. And I just got a text from Julie Pond telling me that their samples in Oregon also have a number of fields with scorch. So we do have scorch present in Whatcom County. And I was struck by Carolyn's slide where she recommended that the BC growers, scout, test, and remove infected plants. And I think that is great advice for us here in Washington. As far as labs, I'm right now aware of 2 labs that should be able to handle, as many samples as we need to send them. And they would be AgDia and the WSU virology lab in Pullman. So I think they have indicated that they're interested in handling scorch and shock samples as well.
Yeah. I think we have one more.
I just want to add to some of the comments that have been made about the importance of managing the vector and managing aphids well. And I work in IPM in BC. So previously, just to share our IPM approach for this, we, in certain fields where maybe a scorch history was not confirmed and where there were no aphids detected pre-bloom during our monitoring, we might not necessarily recommend a pre-bloom aphicide. But at this point in BC, at least, we're kind of shifting towards being very low tolerance, very conservative, and recommending pre-bloom aphicides in all fields, even if 0 aphids are detected. And that's based on data that we've been collecting, showing the increase of aphids throughout bloom, even if zeros were found pre-bloom. So yeah, just to say for BC, where we are really struggling with this scorch virus concern, we're moving in this really conservative direction with our pre-bloom aphicides. And until we have more information, I think that that's sort of the route to go until we really understand what's going on a bit more clearly. So just to add that from BC's perspective.
Sure. Thank you. Thank you for sharing your thoughts. Before we close this panel, I finally want to thank our panel members, Lisa, Caroline, and Dimitar for joining us today. And thank you so much.
Thanks everyone for that panel. Next, we'll be hearing from Alexander Silva, and he'll be talking about calcium accumulation in raspberry fruits to inform fertilizer strategies.
Are you ready to go? Okay. We don't have a right now. Is that okay? Okay.